This is a production of Cornell University.
Okay, so today I'll be talking about genomics and high-throughput phenotyping of cassava brown streak viruses. They told me if I need to point, I need to move. So I'll keep moving, all right? So today we're talking about cassava. So the outline today will be talking about cassava brown streak disease phenotyping, which I'll refer to as CBSD in the presentation. And we'll also be talking about root necrosis image analysis, CBSD viral titer quantification, and also just a summary from the findings of this research. So cassava is an outbreeding species. It's a bushy shrub that's grown for its roots mainly. So, but in Eastern Central Africa, they grow it for also the leaves because they use the leaves to make like stews and pests. So it feeds over 800 million people in the world. And as of 2022, the value of cassava was $4.9 billion in trade around the world. It's propagated by stem cuttings. That's what farmers do mainly. But breeders, we could generate seeds, which are always very difficult to generate. So despite the importance of cassava, in Eastern Central Africa, there's cassava brown streak disease. So this is a viral disease that's very unique in how it expresses its symptoms. That, you could see a healthy plant, but when you uproot the roots, that's like after 12 to 15 months after planting, you will see these patchy necrotic lesions. So this is always very difficult to eat such cassava because the necrosis is hard and you can't boil the cassava and you can't also feed it to animals. So cassava brown streak disease is a positive sense single-stranded RNA virus. It's in the genus Epomovirus and the family Potviridae. You know potato virus Y, which is a very common virus. So these are common plant viruses, but there are two species of this virus, the Uganda cassava brown streak virus, and which I'll refer to as CBSV in the presentation, the cassava brown streak virus and the Uganda cassava brown streak virus. Both viruses cause 70 to 100% yield losses in susceptible varieties. So when we think about the genomics of cassava brown streak virus, just like any other pot virus, it's encoded as one big polyprotein that tends to be cut into different pieces depending on the need, how the plant interacts with, how the virus interacts with the plant. So the big difference with CBSD, which is down, this is the genome of CBSD. The biggest difference between CBSD and other viruses in the Epomovirus genus is this HAM1 protein. So this is a protein that is not encoded by all the other viruses in the Epomovirus genus. And we think that it's because of this protein that it's possible for cassava brown streak viruses to be transmitted by whiteflies and not aphids. So most of the virus in this genome for vector biology, they encode the HC-Pro protein that helps with aphid transmission. So that's the only difference, but most times the proteins are the same. So when we look at the distribution of cassava brown streak disease, this is a very old map from FAO, but I could tell you what's happening. So cassava brown streak disease in the 1930s was discovered in the coastal regions of East Africa. So the virus would only survive at altitude, at very low altitudes, that's below 1,000 meters below sea level, above sea level. So, but as of 2000, the virus moved up to higher altitudes. That has never been reported. And that's why we have the Uganda cassava brown streak virus, because that's where it was discovered in Uganda, while cassava brown streak virus was the original virus that was at the coast of East Africa. So as of last year, we have confirmed cases in the Democratic Republic of Congo, Zambia, and the Central African Republic. So you would wonder why breeders in East Africa are very interested in West Africa. So if you look at the food systems across Africa, West Africa is highly dependent on cassava in their food systems. They don't have alternatives, right? So the alternative could be yam, but everything is largely cassava. So you imagine if cassava brown streak virus moves from East Africa to Western Africa, then we're going to face a lot of food scarcity on the continent. So that's why we're trying to make sure that we breed for it within East Africa and not get it moved to West Africa. So this virus, as I have mentioned, is transmitted by whiteflies. There are many whitefly populations across Africa. But also the greatest transmitters of the disease are farmers because they tend to carry infected planting material from one place to another. So that's how the virus has moved and also evolved to different isolates and different species. So to find cassava brown streak disease in the field, its foliar symptoms are not very threatening. So you see fluorosis along the secondary leaf margins. It's never on the main leaf margin. It's the secondary margins. It's easy to confuse it for, just normal chlorosis sinensis, but with a trained eye, someone could see the leaf patterns and know that this is CBSD. So you also see brown streaks on the stem if you take a good look, but also these also tend to happen on the botanical seeds. So that's a sign of CBSD. So when you uproot the roots, this is always about 12 to 15 months after planting, you see the constriction in the roots, but also when you cut, there are the dark necrotic patches in the root. So those are symptoms of cassava brown streak disease, and they cause up to $100 million in economic losses in East Africa. So when we are scoring cassava brown streak disease, we have this 1 to 5 scoring method that was originally developed for cassava mosaic disease, but also works efficiently for cassava brown streak disease. So a score of 1 means no observable symptoms, while a score of 5 is the highest severity that you can observe, either both on the leaves or on the root. So we tend to score these at 3 months and 6 months after planting for the foliar, because that time the root has not developed. Once you uproot cassava, you can not, you destroy the plant. So we have to wait 12 months to look at the root. So for nearly 10 decades, because I mentioned cassava brown streak disease has been around since the 1930s. So if we count, we've done a lot of breeding for cassava brown streak disease, but we've not identified markers that have been validated in any population. So to my work, we wanted to understand the genetic architecture of cassava brown streak disease, conduct genome-wide association studies for cassava brown streak disease, and explain these results from a breeding perspective. So we're studying a pathogen, but we're also breeders, so we need to explain it from a breeding context. So as John Luke mentioned, the last 10 years, the Next Gen Cassava project has been funding cassava research in Africa. And genomic selection was one tool that, that they promoted for, rapid selection and accelerating of breeding. So we have this genomic selection population that was developed in the first years of the Next Gen project. And, we have it's a SACO2 population. It had 471 clones that were evaluated in the clonal evaluation trials. So when you think about cassava breeding, we have a seedling trial, we have a clonal evaluation trial, advanced yield trial, then the farmer trial. So a clonal evaluation trial is as much diverse as the seedling trial, but we have replication in this case that we don't get in seedling trials. So we planted these trials in 2 locations that have varying CBSD pressure. There's CBSD, but the pressure varies. Then out of these 471 clones, only 320 clones were genotyped. We planted these in an augmented block design with 3 checks, and we used spreader rows within the field to augment the disease pressure. So once we had the population, we had different hypotheses that we wanted to test out how the population performed based on the 1 to 5 scoring method. Then we took root necrosis images because we were curious, is this necrosis due to environmental factors or is it genetic? And also we did viral titer quantification or viral load quantification, and that's how we phenotyped the C2 population. So moving ahead, I will talk about definitions because as someone who works with both breeders and pathologists, it's important to define the context of things that I'll be talking about. So if I talk about tolerance, it means that this is the ability or the capacity of a clone to be infected, but still give satisfactory yield to breeders. When I talk about susceptibility, this clone has the ability to get CBSD, but the disease varies or the expression of symptoms varies. When I mention enhancers of susceptibility, this is the likelihood and the severity of the infection based on the symptoms, right? So it's when it's time to think about viruses, I'll mention that this is based on the virus, but if I mention it anywhere in the presentation, it's due to the symptoms. Then incidence, again, is how frequent these symptoms occur in the population. So let's go to the 1 to 5 scoring method. This paper has been published. So we had the C2 population. We wanted to find out, just understand the population based on the 1 to 5 scoring method. So the research objective is we wanted to evaluate the CBSD trait variability based on the 1 to 5 scoring method within this population, establish phenotypic and genotypic correlations, then conduct GWAS, both univariate and multivariate GWAS, to identify genomic regions that were associated to these traits, and also provide information on functional annotated genes within these regions. So when we talk about GWAS using the 1 to 5 scoring method for cassava brown streak disease, a few studies have been conducted. It's important to mention that Kawuchi's population and Kayondo's population are grandparents of the C2 population. So they did GWAS studies, and they found, Kayondo found that CBSD at 3 months, SNPs on chromosome 11 were associated to CBSD at 3 months after planting, while at 6 months after planting, we only had SNPs on chromosome 4 and chromosome 11. No associations were discovered with the CBSD at 12 months after planting. That's on the root. So the same thing was observed with Kawuchi, that he only picked out CBSD at 6 months after planting, SNPs on chromosome 11. So other studies within East Africa, these are mainly based in Tanzania and Kenya, have done QTL studies with these populations, Namikonga and Chiroba. Just to give you a brief introduction, in the 1930s when CBSD happened, breeders did cross normal cassava, domesticated cassava clones with the wild relatives. And this Namikonga, like a clone like Namikonga or Chiroba, is from that breeding program in the 1930s. So 2 studies have been done using QTL studies, and you can again see that they pick out different genomic regions associated to the different severity scores, either on the folia or on the root symptoms. So that tells you that, some work has been done, but it's really difficult to pinpoint which region in the genome is actually controlling CBSD severity scores. So back to my work, we did want to ask what we were observing in the C2 population due to genetics, environment, or genotype by environment interactions. So we did partition the variants, and you can see that for the C2 population, most of the traits that we measured, CBSD incidence and severity, were highly genetic, while when it comes to genotype by environment interaction, you can see for root severity scores, that G by E is 0, right? So what we see is due to genetic or the environment. So we also looked at variation in broad sense and narrow sense heritability estimates. For broad sense heritability, this is data from the combined data from the different locations. You could see that the estimates generally range from moderate to high, and this is the same with previous published papers for CBSD. And also the same trend is observed with narrow sense heritability estimate, but they tend to be low to moderate depending on the trait. And this is again the same as it's been observed in other studies. So we went ahead to do univariate GWAS analysis. And for our univariate GWAS analysis, you could see that we only picked out associations for CBSD at 3 months after planting on chromosome 1, chromosome 13, and chromosome 18. So as I mentioned, that the ancestors of the C2 population, they picked out SNPs on either chromosome 4 or chromosome 11. In our population, we do not pick out chromosome 4 or chromosome 11. So we went ahead to do multivariate GWAS, but it's important to note that on chromosome 1 and chromosome 18, there's an integration that from the 1930 populations that were developed. So we have integrations on chromosome 1 and chromosome 18. So when we did multivariate GWAS, we wanted to find out which regions, which SNPs we identified in the univariate GWAS were shared among these traits, would show up among these traits. So we again saw that some of these SNPs are shared across, different trait combinations. And because of this, we had confidence to search for candidate genes within these regions to see what type of function do these genes do. So we characterized gene ontologies, which may not be very informative now, but we wanted to cluster them. So based on molecular function, you could see that we have, genes in those regions that control catalytic activity, binding, cellular processes, cellular anatomic entities. So the conclusion from this is, the genes that we received in this case would be enhancers of symptom severity. So if symptom severity, because when you look at, the virus, how the virus proteins work, viruses tend to depend on the host for their replication. So all these help the virus reproduce within the host, and that's how we get the diverse severity scores within the population. So to summarize this section, CBSD incidence in the C2 population, symptom incidence was at 44%. So that shows that while genomic selection, there've been genetic gains from a disease perspective, the disease is still occurring in the population. And we also had a moderate to high broad sense heritability estimates. That's good. Low to moderate narrow sense heritability estimates. 77 functional genes, potential candidate genes were found. The activities again help in symptom severity within the plant. So I'll go to my second objective, which was done with in collaboration with Chris, who used to work for BreedBest in Cornell. So this is the genetic architecture, the genetic analysis of cassava brown streak root necrosis using image analysis and genome-wide association studies. So when we think about root necrosis, if you remember the picture that I showed you of the 1 to 5 scoring method, we score the foliar symptoms twice in a breeding cycle, while root necrosis can only be scored once. So we wanted to expand, how we could characterize or phenotype CBSD root necrosis. So image analysis was the best option that we thought about. So we know that root necrosis is the most devastating CBSD symptoms, because once you have root necrosis, you cannot eat that cassava. And then we thought that image analysis could expand this root necrosis. The question that we had is, we've observed these variations in root necrosis patterns, but were these variations due to genetics or due to environmental factors? That was yet to be found out. So I'll show you what led to our hypothesis of studying root necrosis. You can see that, there are different expression patterns of root necrosis. We agree that, this could be due to, environmental factors. It could also be due to the isolates within the field. So there are so many factors that, lead to these necrotic patterns. So we collected different images and used them for analysis to study root necrosis. So we used PlantCV, which is an open source, software for segmentation of areas of interest in an image. This is a Python-based method. We have the ability to extract features based on size, shape, color, texture. And so we inputted images in PlantCV, and then we got an output file that we use for downstream data analysis. So the experimental design in this case is, we collected 5 roots from each plant in a plot. A plot is made up of 10 plants. So each plant, 5 roots. Images were captured at the proximal end of the root, the middle, and the distal end. Images were collected in both locations across both years. We used tablets with field book to ensure, like, it was a way of controlling the data we got, that if we say this is a plot, then it's an actual plot and an actual image of that plot. Then it's also important to mention that numerous people capture these images. So styles in how they held, the tablets affected our downstream data analysis, since we had to take out some images, since they didn't meet the PlantCV criteria. So we took out numerous images. Again, the research objectives, we wanted to characterize root necrosis image traits, determine phenotypic and genotypic correlations best with the 1 to 5 scoring methods, and also conduct GWAS and also identify, the genes in those regions. So how this happened in image analysis, just to give you, we would input a raw image in PlantCV. Then we use different color spaces for segmentation. So we ended up using the lightness, the green, the magenta, the blue, and yellow, because this was better for us in the segmentation process. Then we would extract the morphological features of the root disc, like in this case, and also in another step, extract the morphological features of the necrosis itself, and we had an output file. So we ended up collecting data, like the traits that we studied were the percentage of necrosis, so the area of necrosis over the whole area of the root disc times 100, the necrotic area fraction, the necrotic Weidish fraction, the solidity of necrosis, and numerous other traits that we studied. So we were curious, what these traits, what's the heritability of these traits? So looking at these traits, all the image analysis traits, the broad sense heritability was generally low to moderate. You can see the percentage of necrosis had 0.42, heritability, while, like the solidity of necrosis or the Kovacs area of necrosis had very low heritability, the lowest heritability estimates. Again, we see the trend, very low heritability and narrow sense heritability estimates associated with these image traits. So we also wanted to find out how these traits really perform against the 1 to 5 scoring method, because those are scores that we're used to and understand best as breeders. So you could see that some traits were highly clustered together. So those are highly correlated traits, while other traits like the solidity of necrosis and the eccentricity of necrosis were negatively correlated with all the other root necrosis image traits. When we compare it to the 1 to 5 scoring method, you can see that CBSD at 12 months after planting has good correlations with all these root necrosis traits that are highly correlated with each other, while the foliar symptoms are not really that correlated with all these other traits. So we also looked at genotype by environment effects on our population, on our image population, and you can see that the Serere 2020 and Amlonge in both years were highly correlated, right? Images from these populations, from these locations were highly correlated. While for the Serere 2019 has, a very different correlation from this. This is because in the Serere 2019, we had a drought at this experiment and lost most of the plants that we had in that field. So the images that we collected were much less. And if you plot this out, you could see that the environment was different from all the other environments. So we went ahead to look at the variations in phenotypic correlations. And you can see again, their traits, just like you saw in the biplot for the genotype by trait interactions, you could see that there's some traits that are highly correlated with each other. And then we have this bunch of traits that, have moderate to low correlations. And then when we look at CBSD severity scores, they have, very poor correlations with all the image analysis traits, except for a few with CBSD at 12 months after planting, but it's still, very little correlations, like the correlation between the percentage of necrosis and CBSD at 12 months after planting is 0.3. So that's a low correlation, but still good. So the same trends were observed with the genotypic correlations, that you could see that there are bunches that, these are highly correlated, these have low to moderate correlations. And then again, we have very poor correlations, but following the same trend like with the phenotypic correlations. So we went ahead to do univariate GWAS, and only 2 SNPs, we found associations with 2 SNPs with 2 traits. The eclipse eccentricity of necrosis, we found SNPs on chromosome 7 and 11, which didn't show up when we did the univariate GWAS analysis with the 1 to 5 scoring method, but has showed up in the ancestors of the C2 population. And then again, we see the percentage of necrosis having SNPs on chromosome R1. So we did multivariate GWAS, and we did not, the different trait combinations, we did not find any significant SNPs with the multivariate GWAS. So again, we looked at the gene functions, and you see again the same trend that follows, that you see catalytic activity, you would see binding, and all these gene functions again help in the enhancing the susceptibility or the expression of the symptoms within the roots. So to summarize this chapter, we know that heritability estimates were much lower than the ones of the 1 to 5 scoring method, because we thought, using images would take away biases that the 1 to 5 scoring method tends to capture, but that didn't happen. We saw similar trends in genotypic and phenotypic correlations. Again, SNPs only associated to 2 traits were identified, and the genes in those regions again enhance symptom expression in CBSD. So that was my objective 2. Going to the objective 3, this was done, so we collaborate with the DSMZ, which is a virus research institute in Germany. So this is the genetic complexity of cassava brown streak disease, insight from qPCR-based viral titer quantification and genome-wide association studies. This work was done in collaboration with Dr. Winter and Dr. Summer, who work at the DSMZ. So when we talk about CBSV titer, the spelling of titer depends on whether you're trained with British English or American English, but it's the same thing. Or viral load, it's just the concentration of the virus within a biological sample. So it depends on, the method that you use. So since we use qPCR, we got CT values that we use to calculate relative amounts of titer within a plant, because we had a reference that we used and used that to detect the amount of virus in our plants. So how this was done is we collected root discs that we compiled together for a plot, like a composite sample. And then those root discs were dried and shipped to the DSMZ, and then the qPCRs were done at the DSMZ, and we got the results back. So again, our objectives for this section was to assess CBSV. These are the two virus species of the disease that we're studying, cassava brown streak disease, CBSV and UCBSV within within the C2 population. We wanted to establish phenotypic and genotypic correlations. Again, do GWAS, univariate GWAS, and provide insights into these genes. So when we look at both viruses in both locations, you could see when we look at the presence, this is plus is presence, minus is absence. When we look at the presence absence of CBSV, we see that CBSV is more prevalent across, the locations and also in the combined data set compared to UCBSV. But we know that, as we planted, from one year to another, there's an increase of CBSV. So to explain what's happening, why CBSV is more predominant than UCBSV, is because some studies have shown that CBSV replicates much faster than UCBSV. Plants have a mechanism of stopping the replication of CBSV, so it may be in the plant, but it just doesn't increase at a fast rate like CBSV does. We also know that the increase from one year to another could be because we replant planting materials. So you imagine if the infection happened in the first year and then you're replanting that, that clone, you're still passing the virus over to the next generation, giving it more time to replicate and the virus to increase. So we also wanted to find out, is virus titer due to genetics, environment, or genotype by environment interactions? So you could see for UCBSV, despite not being very prevalent in the population, it's highly due to genetics. But CBSV, which is more predominant, is due to very little of it is due to genetics, what we're observing. But when we look at genotype by environment interaction, you can see CBSV has high G by E, while UCBSV has low G by E. This could be explained by, again, this is a limitation of our study. We did not test the different isolates and strains within a location where we planted these cassava varieties. So we think that those isolates have different interactions with the genotypes, and that's why we saw high G by E. So moving forward, we recommend that breeders, should periodically test for isolates in these experimental areas to account for those in the analysis. So when we looked at genotype, when we looked at genotypic and phenotypic correlations, again, we see that there are very poor correlations with the 1 to 5 scoring methods, both phenotypic and genotypic correlations. It's important to also mention that CBSV and UCBSV were not correlated. So that brings it to the notion that these are two different viruses acting differently, despite both of them being cassava brown streak viruses. So we did GWAS and we found some regions that were associated to CBSV titer. This was done in GAPIT, that you see chromosome 10, chromosome 13, chromosome 17, and 18 were associated to CBSV titer, while numerous regions in the genome were picked out with the UCBSV titer. Some of these SNPs that we picked really explained, reasonable proportions of variance when we compare it to the 1 to 5 scoring method. So that was a good thing. Again, we looked at the proportion, the genes in those regions of interest. And again, we see these are enhancers of, disease symptoms, as has been observed with all the other phenotypes that we were studying. So we're also curious if we included UCBSV and CBSV titer, viral titer as a covariate for the 1 to 5 scoring method. One, we wanted to improve the precision, but also there's the concept of tolerance, right? Where if you account for titer, then you could pick out regions that control tolerance. So the plant has the virus, but then it gives you good, yield. So we did not pick anything when we used CBSV viral titer as a covariate for the 1 to 5 scoring method. And also the same trend was for UCBSV virus titer as a covariate. Nothing interesting came up. So summarizing this, both CBSV and UCBSV are present in the population, so while we say that the incidence was at 44%, this is confirmed by the qPCR results. We know that CBSV, despite being predominant, is highly controlled by genotype by environment interactions. And that comes to our recommendation that, sampling and characterizing of isolates within locations should be part of routine breeding activities. We identify that there are no correlations between CBSV and UCBSV titer. So that's the implication is breeders moving forward should treat these as two different diseases and develop strategies that are different from each other. So we also noticed that, again, different genomic regions are associated to the trait. That again drives to the point that these are two different viruses that should have two different breeding strategies. So to conclude this talk, we had research-derived variables. Our aim was to characterize the C2 population. We wanted to expand phenotyping. In addition to the 1-5 scoring method, introduce root necrosis image analysis and large-scale virus titer quantification, which we did. So, but based on this, there's no clear-cut method based on the 1 to 5 scoring method that we could recommend for breeders. Breeders have to do a cost-benefit analysis to choose either to do virus titer quantification or image necrosis. But again, more research is needed before they could, make those decisions. And we expanded our knowledge on CBSV titer quantification. So moving forward, as we were talking, you notice different groups tend to identify different genomic regions based on the population that they use, right? So we are proposing that if, the cassava community could do a meta study for all these published papers and have like a baseline, right? That breeders could follow rather than everybody have their own thing, own population. We are also recently the Latin American lines, there are lines that were characterized for CBSV in the greenhouse. And these lines don't, they have differential responses to CBSV, but the most important thing is they don't get infected by either, so you could find a line that's not affected by CBSV, but is highly susceptible to use CBSV. So breeders could integrate this in their breeding populations and, develop resistance, since we've recommended a different breeding strategy for both viruses. So that brings me to an end. I would like to acknowledge, my committee that was John Luke, Michelle Heck, and Keith Perry, the NACRI team in Uganda, the John Luke Yanni Club. We have the people from DSMZ, the Next Gen Project, and the Mueller Lab. Yeah, thank you. I'll take any questions. So for the SNPs that we used were about 37,000 SNPs. DATSIC gave us fewer SNPs, but there's this imputation work that had been done by Manning. So we combined, the former GBS SNPs with the DATSIC to improve the number to get them to 37,000, about that, if I have it right off head. For most, like the 1 to 5 scoring method, and the virus titer quantification, we use the Bufferoni threshold for the GWAS, which is very conservative, but it was good. The root necrosis image analysis, so we did the modified Bufferoni correction threshold, where you calculate a correlation matrix for the number of SNPs on each chromosome, and then use that in calculating the threshold. So it was a more flexible. So it's not the FDR, it's not as less stringent like the false discovery rate, but it's also not very stringent like the Bufferoni correction threshold. So it's in the middle. Yeah, so I tried out, again, this is not like hacking a method. I tried out the FDR and I was getting, again, too many SNPs that, just because it's a low threshold, you get so much that you could not explain or interpret. So I decided to go with the stringent one that I'm very sure that, if a SNP passes that threshold, then something has to be happening in it. So, so again, I think it's because when we're calculating narrow sense heritability, we use the markers. So if we have markers that don't cover like high density markers, so that could be a limitation to why we're getting low narrow sense heritability estimates. So it's still a rough one in my head, but we want because through the next gen, we've generated all this data. So the program at Morag, we have all this data in cassava base. So we want to rerun the GWAS analysis with all these clones that were done and published. And then from that, we could, start talking about what we're seeing, any differences or any similarities that, could be used as a baseline for the population. So it's still a rough idea, but I think standardizing what people use for, determining what's significant in relation with CBSD is important. So the 1 to 5 scoring method has been used. So we have data from since CBSD breeding started. So I apologize for thinking everybody knows that's the method, but across East Africa and Central Africa, the 1 to 5 scoring method is the standard method that's used. So we will do that based on the symptom severity scores from the 1 to 5 scoring method. Image analysis should come in if a breeder feels it would add value to their breeding program, but we're not recommending it for all breeding programs because someone needs to know how to extract the images from plant CV. The analysis part of it is also something that I don't think every breeding program has capacity on. So the 1 to 5 scoring method is understood and will be used. Yeah. Yes. So we took images at the top part of the root. So that's one image, the middle part, and the bottom part of the root. Just the same format that they use when they're scoring cassava brown streak disease. So they'll cut the root, I think, 3 times, and then 3 times, not I think, 3 times, then they'll give an average score of, the score that the CBSD score that they saw at the different places in the root. So that's the same method we used. So each root had 3 images. We had, I think, 150 images per plot. Yeah. So we did not look at that specifically, but we have the data to answer that question if there's variability within the different parts of the plant, and does this vary within the plot and between plots. So we didn't look at, but we have the data to look at it in future. Yeah. Yes, Anna. So there is a whole project on whitefly behavior with cassava brown streak disease. I've not seen any research, any QTL research from that group, but if they do, identify those regions, then it would be possible to look at those regions and see how they, correlate or are the same with the regions that we're picking out. So that's also a limitation of the study. We did not account for whiteflies, but we know that whiteflies did transmit the infection for us. Yeah. Okay, so I think this is Ricky. So for the collection of the categorical disease score, was the data labeled and who collected the data? Ricky, the data was labeled, so we used a field book to collect this data, so we know exactly what the clone is. We also have barcode scanners in the field to help us identify which plant is that. Then the second part of the question is, do you think accounting for RETA effects in your models would improve the results? The answer is yes. We know that breeders have, depending on who's scoring and their experience, they would give a different score to the same plant. So we did not account for RETA biases, but we know that bias exists, and that's why we try to use image analysis to eliminate that bias, but we did not get great results with the image analysis. So Ricky, I hope that has answered your question.